Plasmids are small, circular, double-stranded DNA molecules that can replicate independently of chromosomal DNA. Plasmids carry genes that provide the bacteria with advantages, such as antibiotic resistance. Also, when a bacterium divides, each daughter cell receives a copy of the plasmid contained within the mother cell. Plasmids' most important characteristics are that they are extrachromosomal DNA, and they carry functions advantages to the host. They have other several characteristics such as size, copy number, function, and their capacity to be transferred. Plasmids can be of a variety of lengths, from 1,000 base pairs to hundreds of thousands of base pairs. The copy number influences the strength of the plasmid characteristics, especially antibiotic resistance. There are also different plasmid types with different functions, like R plasmids, F plasmids, killer plasmids, degradative plasmids, and virulence plasmids, which have the functions of antibiotic resistance, promoting conjugal transference of genetic material, synthesis of toxins that kill bacteria, synthesis of degradative compounds, and introduction of tumors. Some plasmids possess the characteristic of being conjugative, meaning they can be transferred through bacterial conjugation. Finally, all plasmids are self-replicating, and many copies of them can be made by growing the plasmid in bacteria. One of the most common characteristics is resistant to a certain antibiotic, like ampicillin. Plasmids could be high copy or low copy, which allows the microorganism to have characteristic of expressing high or low quantity of determinant genes. Plasmids can also provide the microorganism with virulence, therefore a non-virulent microorganism can become virulent. A microorganism can also become resistant to heavy metal after a new plasmid acquisition. Plasmids could also be responsible of making microorganisms able to survive in presence of alternative and non-common carbon sources, as well as providing microorganisms with the ability of creating symbiosis. A microorganism can also acquire resistance to a certain bacteriophage by getting a new plasmid. They too can gain a certain characteristic that could allow them to colonize certain tissues in a host organism. There are plasmids that give microorganisms the characteristic of forming tumors in plants. Finally, getting a new plasmid can allow the microorganism to change color in presence of some reagents. There are some basic steps that must be included in any plasmid purification, which are cell growth and harvesting, cell pellet resuspension, lysis, and neutralization, and final steps that consist on cleaning up the sample from salt excess, RNAs, or buffer residues. There are some differences between the extraction protocol of a plasmid and another acid, nucleic acid. The methodologies for plasmid extraction are different than the ones for genomic DNA or RNA extraction. In genomic DNA extraction, cells must be lysed to expose the DNA, and the cell membrane should be removed using an appropriate detergent. In this protocol, proteins and RNA can be removed by using RNases, proteases, and phenylchloroform isoamyl alcohol, which denatures proteins. By using the phenylchloroform isoamyl alcohol solutions, proteins, lipids, carbohydrates, and cell debris are removed through the extraction of the aqueous phase from the organic mixture. The aqueous phase contains DNA that can be precipitated with ethanol. Alkaline lysis is a method used in molecular biology to isolate plasmidic DNA from small volumes of bacteria. This method is based on the principle of differential renaturation of chromosomal and plasmid DNA in order to separate it. It involves three basic steps: lysis of cell, separation of plasmidic DNA from chromosomal DNA, and purification of plasmidic DNA. One of the most used methods is the mini-prep method, which uses step, buffer, and lysosome to lyse the cells. After lysis, the DNA is treated with a phenylchloroform mixture and then precipitated with isopropanol, thus being free from impurities. Lysosome is an enzyme used to break down bacterial cell walls. Lysosomes are a family of enzymes with antimicrobial activity. In electrophoresis, DNA fragments migrate according to their length, but only if all the molecules have the same form. When plasmidic DNA is extracted, the plasmids can have 2 or 3 different forms, so in the electrophoresis shells, there can be seen 2 or 3 bands. These bands correspond to the 3 possible isoforms of the plasmid: supercoiled, open, circular, relaxed, and linear. The difference between the bands is due to the difference of volume that each isoform occupies.
1.5 milliliters of the culture was centrifuged for 20 seconds at high speed in order to obtain a bacterial pellet. Then 100 microliters of the GTE solution were added in order to resuspend completely and correctly the pellet. Then the solution was complemented with lysozyme and mixed with the vortex. Also 200 microliters of SDS alkaline solution and 150 microliters of high salt solutions were added. After centrifuge, the supernatant was transferred to a new microtube. Then, in order to precipitate the plasmid, 2 volumes of cold absolute ethanol were added. After centrifuge, water was added to dissolve the pellet. In the electrophoresis gel, the marker bands are seen clearly. Also, there are degradation and some other different contamination that is shown with the blurred lines and the blots formed at the top and at the bottom of the gel. Also, there are shown lines at the sides of the lanes all along the gel. And finally, we can see the different bands of the isoforms of the plasmid. The bands of the marker are defined correctly, but the separation from one to another is not the correct one. The different blurred lines shown from the well to the first band shown in the gel can be because there is contamination of proteins and also because the plasmid could be degraded during the electrophoresis. The contamination shown can be by proteins in the well, degradation seen in the blood lines, and also the blots shown in the electrophoresis gel in the middle and in the bottom can be because of the presence of the RNA as a contaminant. Another factor that could influence directly in the run of the electrophoresis gel can be the lysozyme used in the protocol. This can be because of the conditions of the lysozyme need to be the correct ones and need to respect the time of the action of it. We can see in the lanes that there are little lanes at the sides, and this can be because the gel was damaged because of incorrect use of the micropipette and the tips.